right, up next we have Sahar Abubucker from Chroma Medicine, who's going to tell us more about epigenetic editing and how multiplexing with this can avoid some of the large unwanted chromosomal changes that we're all terrified of with nucleases.
Thanks for the introduction, Ben. And thanks to the organizers for this opportunity to share some of our work at Chroma Medicine, where we'll focus on harnessing epigenetics, the cell's innate system for gene regulation, to create transformative therapies for patients by modulating expression of one or more genes without cutting, nicking, or causing any changes to the DNA sequence. So epigenetic editing leverages the cell's endogenous machinery that it uses to regulate gene expression. And epigenetics regulates gene expression by controlling how DNA is packaged. The aspect of epigenetics that we have focused on at Chroma is DNA methylation. When DNA is hypomethylated, it is open and accessible, allowing transcription factors to bind and drive gene expression. When we want to turn off a gene, we transiently apply an epigenetic repressor that methylates the target locus, driving the DNA to a closed and inaccessible state, thereby silencing the gene. And this is done without any cutting, nicking, or alterations to the DNA sequence, thereby driving a durable change in phenotype without a change in genotype. In today's talk, I'm going to share data demonstrating that epigenetic editors are durable and have high efficacy. And it does this without any inherent consequences seen with cutting and nicking, which you see with other editing approaches. So I'm going to first describe Chroma's epigenetic editing construct. Early proof of concept data generated by in the labs of Angelo Lombardo and Luigi Naldini, two of our founders, showed that targeted methylation can durably silence a gene. Chroma's epigenetic editing construct is based on subsequent work by two of our other founders, Luke Gilbert and Jonathan Weisman, and is a single fusion protein with 3 functional domains. The transcription effector domain transiently represses the target gene. The methylation effector domain durably silences the target gene by methylating the locus. And the DNA binding domain is the one that precisely localizes these effector domains to the target sequence. The DNA binding domain used in Chroma's epigenetic editor is a dCas9 protein. Most of you are probably very familiar with dCas9. It is a nuclease inactive version of the Cas9 protein. And the 2 domains in Cas9 that confer nuclease activity have been characterized. And in the dCas9 version, those 2 domains have been inactivated through mutations. So in the next few slides, I'm going to share data showing that the epigenetic editor has no nuclease activity and does not cause any double-stranded breaks, and therefore there are no indels, translocations, or any other type of genomic rearrangement events. But first, I want to highlight the efficacy of Chroma's epigenetic editing platform. So here's data that was shared by Aaron Jaffe, our head of research at Chroma, yesterday. And this is data showing durable and efficient silencing in 2 different therapeutically relevant loci in vivo. On the left is PCSK9, on the right is HBV. And the epigenetic editor is represented in the blue bar, which is pretty close to the x-axis. Aaron had a chance to talk about how these epigenetic editors have very high specificity, but did not get a chance to share data on the lack of nuclease activity, which I'm going to share today. So using the same epigenetic editor that was used in vivo, we repeated the experiment in vitro, and we used those cells to test for indels. Now, indels are formed when DNA repair pathways are triggered by double-stranded breaks to the genome. And we used indels are what happens when you have a Cas9 nuclease. We used a deep amplicon sequencing assay to look for indels using epigenetic editors as well as Cas9 nuclease. We also included untreated cells as controls. We examined 2 different targeting sites. Cas9 nuclease must target the coding region, whereas the epigenetic editor prefers the regulatory region. With both these methods, we had very high silencing of PCSK9, which is shown in the plot on the left. And so the colors, the gray is the untreated control, blue is the epigenetic editor, yellow is the Cas9 nuclease. And if you look at the percentage of indels reported on the plot on the right side, we see that the only time we see indels are with the Cas9 treated cells at the Cas9 editing site, which is what we expect. In contrast, the epigenetic editor is at the rate of the controls. Now, this is really exciting because the possibility of durably silencing a single gene without causing any cuts to the genome opens up the possibility of multiplexing, where you won't have any consequences of making multiple cuts. And one of the applications of multiplexed editing is in cell therapies. And so we wanted to next ask the question whether we can apply this technology in primary cells. So here is data showing durable silencing of a single target in primary human T cells. This is, the yellow bars are CRISPR-I, which is very similar to the epigenetic editor, but lacks the methyltransferase domain. And the gray bars are the untreated controls, and the epigenetic editor is represented in the blue bar close to the x-axis, showing durable and efficient silencing all the way to day 36, and this was maintained through a strong restimulation. We next wanted to ask whether we can use this approach to do multiplex silencing. So here is data with the epigenetic editor on 3 different targets. So the gray bars, again, represent the untreated controls, and the blue bars are the epigenetically edited cells. We see very high silencing of all the 3 targets. So we've already seen that there are no indels with epigenetic editing. So we next wanted to ask, are there other types of chromosomal rearrangements like translocations and other events that are caused with the epigenetic editor? So for this assay, we used a unidirectional sequencing assay that can detect translocations in addition to indels. And we also looked at an imaging assay which can broadly look at different types of chromosomal rearrangement events. So here is data from the unidirectional sequencing assay. So in terms of indels, we see similar rates of indels that we saw in the previous plot, where we only see indels with the Cas9 treated cells at the Cas9 site. The epigenetic editor does not have any indels. And if you look at the plot on the right, this is just showing translocations between 2 genes. We see a rate of about 3% between 2 genes, and when we include translocations between all the 3 genes, we see a rate of about 7%. And this is very similar to what's been published before and shown in other studies. So sequencing shows us that there are no indels or translocations with the epigenetic editor. But sequencing can miss many potential outcomes, and so we wanted to look at a imaging-based approach that can capture other types of events like translocations, chromothripsis, losses, gains, etc. And so to do this, we used a targeted FISH assay, which is a chromatid insight assay. And I have a picture on the right which shows what that an image looks like of a single cell. This is a normal cell. So we have probes targeting the 3 target genes. Each probe is represented in a different color. And you can kind of see that for every gene, you see 2 dots, which is representing the gene in the pair of the chromosome. And in a normal cell, all these 3 genes are in different chromosomes. So for this experiment, we also included a nucleofection control. So we used an epigenetic editor with a non-targeting guide, so it does not have any match to the genome. So using this approach, I'm showing you a picture on the right, which is what a translocation looks like with this imaging assay. So essentially, you see co-localization of probes that target different genes on the same chromosome, indicative of a translocation. And I'm just summarizing the total number of translocations on the plot on the left. And similar to the sequencing assay, this targeted FISH assay shows a very similar rate of translocations. But in addition to translocations, I mentioned that this imaging assay can also look at other types of chromosomal rearrangement events. And so I've highlighted 2 different examples on the right side. So the top image is an edit site truncation with the Cas9 nuclease, where you see that the rest of the chromosome is truncated at the edit site. And the image on the bottom shows a chromothripsis event. And chromothripsis is when you have a massive rearrangement event of the genome, of the chromosome. And this looks like the chromosome is kind of shattered. So if you look at the total number of events, which is summarized on the plot on the left, we see that with the Cas9 nuclease, we see almost a third of the cells showing some sort of a chromosomal rearrangement event. And in contrast, the epigenetic editor is similar to the controls. So in summary, epigenetic editing leverages an endogenous system used by cells to regulate expression of a single or multiple genes. And it can durably silence multiple genes without altering the DNA sequence. And Chroma's epigenetic editors can efficiently multiplex without causing any indels, translocations, or other genomic rearrangements. And this makes it really well-suited for simultaneous multiplexing. So with that, I want to extend a huge thank you to the entire Chroma team and collaborators, and our collaborators at Chromatid. I'm happy to take any questions.
Thank you. That was an awesome talk. 10% of cells naturally have chromosomal aberrations?
Yeah, thanks for the question. That was something that was surprising to us as well. So in talks with our collaborators at Chromatid, I think it's a consequence of the cell type that's being used as well as the delivery method. So this is nucleofection, and we're trying to do these experiments to determine what percentage of those are caused by the delivery method. But there are some events that occur on a normal cell at a low frequency, like cystochromatid exchange events, for example.
Okay. Over here.
I just wonder, is there any mechanism by which epigenetic editing will be driving chromosomal translocations that I'm not aware of? So what I see you spend so much time showing these, improving these, which is awesome, but just wondering if there is something I'm missing.
Yeah, we are developing therapeutics, and so we want to be extremely sure that there's no sort of genomic rearrangement events. And you can see that a lot of types of like cutting and nicking events, there are unintended chromosomal rearrangements, and so we wanted to be doubly sure that we don't have any double-stranded breaks and we're not causing any sort of chromosomal rearrangements.
I was just wondering, maybe it was like a leftover nickase activity or something on the dead Cas9, something like this, a low activity. So good.
Yeah.
That.
Exactly. So there isn't a lot of published data out there that have looked really deeply sequenced data to make sure that there are no double-stranded breaks, and so we wanted to be able to do that.
Awesome. And another question, if I may, how many guide RNAs do you usually need to achieve such a great silencing?
Yeah, great question. So we spend a lot of time and effort at Chroma to really comprehensively screen guide RNAs and effectors to make sure that we have highly efficient silencing. So, I showed data in vivo data showing extremely durable silencing. So with our technology, you can use multiple guides to target different loci. You can also use multiple guides to target the same loci. But based on our experience, what we've seen is that, single guides are usually enough to really have durable silencing.
Nice. Thank you.
Thanks.
We'll go in the middle here.
There are one more.
We'll come back. Go.
Yeah, okay. So you are using DNMT3A and 3L. Both are robust and context-dependent methyl transferases, right? And you are combining them with the CRAB. So do you check the global guide RNA independent methylation in these cells?
Do we see global methylation in other
guide
places?
RNA independent. If you deliver on the AP editor, check the global methylation pattern.
Yeah, so the data published by our founders, has looked at that extensively, and we've also looked at it extensively. And Aaron actually showed some of that data yesterday, looking for expression and methylation. So we don't see any changes in global events. It looks exactly the same, except at the intended target locus. And we spent a lot of effort to make sure that's the case.
And have you, second question related, have you found a window where actually these epigenetic editors are doing the methylations?
So yes, so it can be very context dependent, depending on the gene and what the context of the methylation is at the locus. So it's on a gene by gene basis. But the methylation is pretty targeted, and it does not spread like widely. It's limited to a specific window.
Okay, thank you.
Yeah, thanks.
And over here.
Yeah, thank you for the great talk. I think it's a follow-on question. I was very curious why you were looking at chromosomal rearrangements for an epigenetic editing, because I would be more concerned about which regions are suddenly becoming closed in comparison to being open as off targets. So yes, as you mentioned yesterday, I did attend the talk and I saw that there was no change in the global profile, but I was actually curious, how long did you follow that on in terms of concluding that there are no off target related to that?
Yeah, so I showed data from day 3. We've also done day 7. So the methyl, the epigenetic editor is applied transiently, and so it does go away after a period of time. It's not permanent expression. So we haven't done a time point after day 7, but we see very high silencing at day 3. So we don't expect anything, but That's a fair question.
Yeah, no, I think my point was in terms of understanding the gene expression, because some genes,
okay.
yeah,
Oh,
ultimately.
sorry, I misunderstood.
Yeah.
Yes, so for profiling specificity, which we have an extremely comprehensive set of assays that we use to profile specificity, and we do that at a time point when the epigenetic editor is no longer present. So we use a very late time point based on the different cell systems we're using. It's at a fairly late, like day 14, or at a time point when the epigenetic editor is no longer present in the cells.
Thank you.
I think we have time for one more. So if you guys want to rock, paper, scissors and can go.
Hi, Marco Alessandrini from Antion Biosciences. You achieve very efficient silencing in the order of 70 to 80% for all your targets, which I assume is functional. But can you achieve knockout status with this approach? Or is that the ceiling, do you think?
Thanks for the question. So the in vivo data that I showed, we had super high silencing at 99%. The cells that we used for doing this analysis was some of our early guides where we hadn't actually done the full screening, so which is why we only had 80%. But typically, this was just mainly to generate this data set. But once we've done the full screening, we see very, very high efficiency.
Okay, good. Thank you.
Thanks.
All right, let's thank Sahar again.